Right, so the next thing that we actually have to look at is polysaccharides. Now, if you look at the definition, always remember that saccharides means sugar and poly means many. If you remember, monosaccharides means one sugar molecule, disaccharides means two sugar molecules linked together, and polysaccharides, by definition, is just many repeating sugar units linked together by glycosidic bonds. Now, so let's look at an example with alpha glucose. So remember, alpha glucose is a monosaccharide. The reason why it's a monosaccharide is because it is a one sugar molecule. It's just made up of one sugar. And always get used to drawing out the ring structure of alpha glucose. Remember that for alpha glucose in carbon number 1, the OH is pointing downwards, and also carbon number 4, the OH is also pointing downwards. Now, so that's one alpha glucose. But if we have two alpha glucose molecules right now, as you can see over there, if a condensation reaction happens where you remove a water molecule and you link together the two alpha glucose molecules together, it forms something called a glycosidic bond. Now, this is known as a maltose, and maltose is referred to as a disaccharide. The reason why it has to be referred to as a disaccharide is because, well, it's just basically made up of two monosaccharides linked together by a glycosidic bond. What happens if we keep on doing this chain? We keep on doing this pattern. And when we keep on doing this pattern where many alpha glucose are linked together repeatedly, that's when we get something known as a polysaccharide. And as you can see, the polysaccharide has many glycosidic bonds and many alpha glucose molecules. You may be wondering, if mono is 1, poly is 2, how many is poly then? Poly is usually referred to when it's more than 20 alpha glucose linked together. That's basically it. But we don't have to know that. As long as you put the word many repeating units of sugar in the exam, you're good. So the first polysaccharide that we are looking at is starch. Now, starch is basically an energy storage molecule in plant cells. Now, what do I mean by energy storage molecules in plant cells? To understand why starch is referred to as an energy storage, we have to look at a situation. Now, in this situation here, I'm just drawing out a plant cell. Now, always get used to drawing a plant cell. I'm just putting in the cytoplasm. And if we zoom in, we have a chloroplast right there. Remember chapter 1, how do we recognize that that's a chloroplast? You can see the structures known as thylakoid. You can see the double membrane organelle. Chloroplast is a double membrane organelle. I'm just drawing out one chloroplast. And within the chloroplast, those purple colored dots, we just assume that those purple colored dots are glucose molecules or alpha glucose molecules. You see, once alpha glucose is synthesized by chloroplasts through the process of photosynthesis, the alpha glucose is small enough to diffuse out of the chloroplast membrane. And once it diffuses out of the chloroplast, it can then be used by the mitochondrion for a process known as aerobic respiration. So you see, sometimes when the chloroplast actually synthesizes a lot of glucose molecules, however, they don't necessarily have to use all the glucose immediately. Some of the glucose can be kept for the future. But the problem here is, because glucose is small enough, it may diffuse out of the chloroplast, all right, and it might also diffuse out of the cell, and we don't want that to happen. Another big problem is glucose as a molecule is soluble in water. And because it's soluble in water, it can actually lower the water potential of the chloroplast. And this causes water to rush in by osmosis. And if water rushes in by osmosis into the chloroplast may burst. And that's a problem. The reason is because glucose is able to affect the water potential within the chloroplast. Because glucose is a soluble and it's a reactive molecule. So these are the two problems. It can affect the water potential of the cells, and it can also escape or diffuse out of the cells. So the solution to this is very simple. Instead of just basically keeping them or storing them as glucose molecules, chloroplasts will basically just link the glucose molecules together. And when they link the glucose molecules together, just like those dots over there, when they link it together, that's when they get something known as starch. And by linking the glucose molecules together, it makes the size of the molecule larger. And when it makes the size of the molecules larger, this is good for two reasons. Number one, the molecule is too large to diffuse out of the chloroplast. Therefore, it cannot diffuse out of the cell. Number two, it will not affect the water potential of the cell. The reason why it doesn't affect the water potential of the cell is because starch as a molecule is insoluble in water. So those are the two reasons. It does not affect the water potential. It's not able to diffuse out of the cells. So that's basically why plants convert glucose into starch. So coming back to starch, which is the energy storage molecule in plant cells, there are two types of starch molecules that we have to know. The first type of starch is known as amylose, and amylose is made up of alpha glucose molecules, but they only contain 1,4-glycosidic bonds only. So what I'm going to do is I'm just basically going to throw out a few alpha glucose molecules. And as you can see, when one alpha glucose molecule links with another alpha glucose molecules, they form the glycosidic bonds at a certain angle. There's a reason why they form the angle, but we don't have to know why they form it at an angle. We just have to know that when they form the 1,4-glycosidic bonds, which I'm putting it in red over there, I hope you can see it, they form it at an angle. So what happens is the molecule, the chain, starts to curve a little bit. So the reason why this is important is because if you have many repeating units of glucose molecules linked together, as you can see here, they are just basically starting to coil because of that 1,4-glycosidic bonds, which causes the shape of amylose to become a helical or spiral shape. That is why amylose is referred to as a helical molecule, a coiled molecule, or a spiral molecule. And that's a polysaccharide. The second type of starch molecule that we have to know is something known as amylopectin. Same as amylose, amylopectin is actually also made up of alpha glucose molecules. The difference here is they contain alpha 1,4 and also alpha 1,6 glycosidic bonds, but they are not helical. So as you can see here, the red color bonds are the alpha 1,4 bonds, but the green color ones are alpha 1,6 bonds instead. So if the molecule is only predominantly made up of alpha 1,4 bonds, if it's only made up of alpha 1,4 bonds, they form a coil. But because they have alpha 1,6, however, they start to form a kind of branched shape. So the green color lines signify the alpha 1,6 bonds and the red color lines signify the alpha 1,4 bonds. And you can see in the alpha 1,4 bonds, they are curling or they are kind of coiling, or they're trying to coil. But with the presence of 1,6 bonds, it causes amylopectin to become branched. That's basically it. And of course, the second type of polysaccharide that we have to look at is glycogen. Starch is an energy storage molecule in plant cells. Glycogen is an energy storage molecule in animal cells. In terms of structure, glycogen is again made up of alpha glucose molecules. It contains alpha 1,4 and also alpha 1,6 glycosidic bonds. So if you notice, it's kind of the same as amylopectin in plants. The main difference is, even though it's branched, it's just basically more branched than amylopectin. If a question asks you, what is the main difference between glycogen and amylopectin, if you wanted to answer that, you can say that glycogen contains more alpha 1,6 bonds because it's the alpha 1,6 bonds that causes it to be branched. So that's it. So I've already mentioned it over there. So as you can see, these two cells over here, just as a bit of a, this is just basically the animal cell. If they have too many glucose molecules inside the animal cells, it will lower the water potential. And what will happen to the animal cells? The water will start rushing into the animal cells by osmosis. And it causes your animal cells to burst. Okay, our cells to burst basically. Because remember, we are, at the end of the day, we are animals as well. So our cells will burst when we have too much glucose molecules inside the cytoplasm. However, if your cell converts it into glycogen, it will not affect the water potential inside the cell. It will equalize the water potential inside and outside the cell, so no osmosis will take place. And there you go. Your cell is fine. That is the main reason why animal cells and plant cells convert glucose into polysaccharides, such as starch or glycogen. These are the main reasons, and that's why they're known as energy storage molecules. And of course, when the plants or animals need extra glucose, what they can just do is they can just break down the starch or break down the glycogen and use up the glucose when it is actually necessary. Right. The the third type of polysaccharide that we are going to be looking at is cellulose. We've seen starch, we've seen glycogen. Cellulose is actually the third polysaccharide that you have to know for the A-level syllabus. The difference between cellulose and starch and glycogen is that cellulose is actually made up of beta-glucose molecules, and they are joined together by beta-1,4-glycosidic bonds. So as a revision, let's just do a comparison between alpha glucose and beta glucose molecules. If you remember alpha glucose, they will have the OH groups for carbon number 1 and carbon number 4 facing downwards, but for beta glucose, the OH group in carbon number 1 is facing upwards. So even though it may look like a small difference between alpha glucose and beta glucose, the consequence of this difference is quite significant. And what do I mean by that? Let's look at this. So I told you that to polymerize beta-glucose molecules together involves a process known as condensation. So what we have right now is we have two beta-glucose molecules. The problem right here is they cannot form the beta-1,4-glycosidic bonds. The reason is because, as you can see, the OH groups are quite far away. So the solution to this is pretty simple. The second beta-glucose molecule will have to rotate 180 degrees, and if you see me rotating it, let me just change it up a little bit. And once it has been rotated 180 degrees, look at the OH group between carbon number 1 and carbon number 4. The OH groups are quite close to each other. Right now, can condensation happen? Yes, it can happen because the hydroxyl groups or the OH groups are quite nearby. So once condensation happens, this is where you have a beta-1,4-glycosidic bond. If I were to put another beta-glucose molecule next to it, does that other beta-glucose molecule have to rotate 180 degrees? It doesn't have to rotate 180 degrees because the OH groups are quite close to each other. So that's okay. But the fourth beta-glucose molecule will have to rotate again. The reason why it has to rotate is because it wants to make the OH groups quite close by. So what we notice over here is, if I were to just basically draw out a few beta-glucose molecules, not every beta-glucose molecules has to rotate 180 degrees. And this is when we get something known as a cellulose molecule, and every other beta-glucose molecule rotates 180 degrees. And every other beta-glucose molecules are the ones I'm highlighting in yellow. So only those in yellow had to rotate. And once you link many beta-glucose molecules together, that's when you get something known as cellulose. The shape of cellulose is going to be a bit of an interesting one. But before we look at cellulose, let's look back at amylose. If you remember amylose, which is a type of starch, amylose was basically made up of many alpha glucose molecules. And when you have all the alpha glucose molecules linked together, they kind of start to curve, because remember I said when they form alpha glucose molecules, they form it at an angle. And when the chain starts to coil, it will cause amylose to become a helical, coiled, or spiral shape. That's what we saw. But cellulose, however, is a little bit odd. If you notice what's happening here, I'm just basically drawing out many beta-glucose molecules and many alpha-glucose molecules linked together. Because every other beta-glucose molecule rotates 180 degrees, the glycosidic bond does not curve. You see, because the glycosidic bonds are not all facing one direction. Some glycosidic bonds face upwards, some glycosidic bonds face downwards. So what happens is, this just basically neutralizes the angle. It doesn't curve or it doesn't coil in any way whatsoever. It just basically makes beta glucose become a rather linear shape. And as I'm highlighting it here, notice the glycosidic bonds where I'm highlighting it in green. Okay, it does not coil. It just basically forms a linear structure. So because if you look at the alpha glucose, their glycosidic bonds are orientating downwards, it causes the shape of the chain to coil or form a helix. But in beta glucose molecules, some glycosidic bonds orientate upwards or face upwards, some glycosidic bonds face downwards. Therefore, the beta glucose molecule does not coil and it forms a linear structure. That's what we just have to know. You don't have to know why it forms a linear structure. You just have to know in the exam that cellulose molecules are just linear, amylose are helical, even though both of them have 1,4 glycosidic bonds. The main difference is amylose has alpha 1,4 glycosidic bonds, beta glucose has beta 1,4 glycosidic bonds. So why is it a big deal that cellulose is a linear molecule? The main reason is because if we were to look at this in detail, what I'm doing is I'm just drawing out one cellulose molecule as a green line, and I'm just putting it over there, and another cellulose molecule down there. Interestingly, between one cellulose molecule and another cellulose molecule, they can start to form something called hydrogen bonds. So what exactly are hydrogen bonds then? Hydrogen bonds form between atoms with partially positive charges and partially negative charges. I will be talking about hydrogen bond in the next video, so you don't have to worry about that. But right now, we just have to know that if an atom has a partially positive charge and another atom has a partially negative charge, they can form something known as a hydrogen bond, where I've signified in a purple colored dotted line. So let's just draw out one cellulose chain right here. Oxygen, what we just have to understand here is oxygen has a partially negative charge and hydrogen has a partially positive charge. We will talk about the reason why oxygen has a partially negative charge and hydrogen has a partially positive charge. I will cover it in the next video, hopefully. But for now, what we just have to know is oxygen has a slightly or partially negative charge and hydrogen is partially positive. And because they have this difference in charges, what I'm doing here is I have two cellulose molecules, one at the top and one at the bottom. Notice that I've circled the hydrogen at the top and the oxygen at the bottom line, and they can link together and form something known as hydrogen bonds. So individual hydrogen bonds can actually form between hydrogen and oxygen. Individually, one hydrogen bond is weak, but cumulatively they become stronger. So the more hydrogen bonds link up between one cellulose and another, it makes it stronger. So why is this a big deal? So as we can see here, one cellulose and another cellulose can form many hydrogen bonds with each other. So the reason, because it's linear and because they can form hydrogen bonds, many cellulose molecules then can be tightly packed together and they can also bond with each other. So what I'm drawing over here are just, each of those lines are just individual cellulose molecules packed tightly and they form a kind of bundle. And that bundle over there is known as a cellulose microfibril, which is made up of a bundle of about 60 to 70 cellulose molecules linked together by hydrogen bonds. And those hydrogen bonds, if you notice, I'm drawing it in purple right there. So they can be packed into a very small area because their shape is linear. You can imagine one cellulose molecule to be like a stick, and you can imagine cellulose microfibrils to be a bundle of sticks. So if I were to ask you a question, which is stronger? Obviously, you're going to say the bundle of sticks is stronger because they're all grouped together. It's easy for you to break one stick, but if I ask you to break a bundle of sticks, it's going to be difficult. And what happens is many of the cellulose microfibrils can then also group together by more hydrogen bonds, and they can then form something known as cellulose fibers. So cellulose fibers are just like many bundles of sticks stacked together. So they have a very high tensile strength. They do not break easily. They do not bend easily. They do not stretch easily. The reason why this is significant is because cellulose fibers are actually used to make plant cell wall. And if you remember, plant cell wall are extremely rigid and they can actually withstand a high pressure when water rushes into the cell. The plant cell will not burst because the cell wall is strong enough to actually withstand the pressure. So this is why cellulose molecules being linear is a big deal. Because they are linear, they can actually be packed together in a small space where you can pack a lot of them together to form something called microfibrils. And microfibrils can have hydrogen bonds, okay? And many microfibrils group together to become cellulose fibers, which are much stronger. And these cellulose fibers are then used to make the plant cell wall. So to summarize the whole thing, if we wanted to look at the three types of polysaccharides that we have covered, the first polysaccharide that we looked at was starch, which is basically the energy storage in plant cells. And starch is divided into amylose and amylopectin. Amylose is made up of alpha 1,4 glycosidic bonds and the shape is helical or spiral. Amylopectin, however, is made up of alpha 1,4 and also alpha 1,6 glycosidic bonds, and the shape is branched. The second polysaccharide we looked at was glycogen, which is the energy storage in animal cells. And the shape glycogen is actually made up of alpha 1,4 and also alpha 1,6 glycosidic bonds, and their shape is also branched. The difference is with amylopectin is glycogen has more alpha 1,6 glycosidic bonds. Therefore, they are more branched. The third polysaccharide we then looked at was cellulose. Cellulose is made up of beta 1,4 glycosidic bonds, which means to say they are made up of beta glucose molecules. And the shape of cellulose is linear. That is the summary for polysaccharides.